Assalamualaikum and hi. My name is Alya Batrisya Binti Mohammad. My MATRIC number is E2382582. I am from SES Year 2. So today, I want to present about my mini project. The title of my mini project is Water Quality Assessment in Pulau Tanjung, Tumpat, Kelantan using Algoremediation process. Water quality plays a crucial role in maintaining ecological balance and supporting the health of local communities, particularly in rural and developing areas. Effective water quality assessment is essential for understanding the physical, chemical, and biological characteristics of aquatic system. Parameters such as temperature, turbidity, pH, dissolved oxygen, nitrate, and phosphate concentration are commonly monitored to evaluate the condition of water bodies and identify potential sources of pollution. In Malaysia, Pulau Tanjung, located in Tumpat, Kelantan, is a developing area with emerging socio-economic and tourism activities. The island has become a popular destination due to its vibrant local market, which attracts a significant number of visitors. However, this increased human activity has contributed to growing environmental pressures, particularly in terms of water pollution. Improper waste disposal, agricultural runoff, and wastewater emissions from nearby settlements pose significant threats to the island's aquatic ecosystem. Traditional wastewater treatment methods such as chemical coagulation, filtration, sedimentation, and chlorination are widely used. Although effective, these systems are often costly, energy-intensive, and may result in the production of secondary pollutants. As an alternative, biological treatment methods have gained attention for being more sustainable and environmentally friendly. One promising approach is algoremediation, a process that utilizes algae to remove contaminants from water through absorption, assimilation, or transformation. Algae such as Chlorella vulgaris have demonstrated significant potential in nutrient removal and water purification due to their high growth rate and ability to assimilate nitrogen and phosphorus compounds. Chlorella vulgaris, a unicellular green microalgae, has been widely recognized for its effectiveness in bioremediation due to its capacity to absorb excess nutrients and improve dissolved oxygen levels. The application of this species is algoremediation is considered cost-effective and energy efficient compared to conventional methods. Despite its potential, there is limited research focused on the application of algoremediation in Pulau Tanjung, particularly concerning its effectiveness in improving local water quality. The present study aims to assess the current water quality status of Pulau Tanjung and evaluate the effectiveness of Chlorella vulgaris in improving water quality through algoremediation. The Water Quality Index (WQI) and the National Water Quality Standards (NWQS) for Malaysia are used as reference frameworks to classify and interpret the water quality data. This research is timely and relevant as it contributes to the limited body of knowledge on the ecological conditions of Pulau Tanjung and explores the viability of algoremediation as a sustainable solution for water pollution. We've moved to problem statement. Most rural coastal villages and towns, including Pulau Tanjung Tumpat, Kelantan, have deteriorating water quality. Major issues such as turbidity, nutrient pollution, and potential contaminants have been brought on by the discharge of contaminants from wastewater into nearby water bodies. The contributing factors for the issue are largely anthropogenic activity, agricultural runoff, and others. Along with human well-being, aquatic and agricultural activities are also affected adversely due to diminishing water quality. Despite the significance of this issue, not much research has been conducted to assess Pulau Tanjung's water quality at the moment and look into natural, sustainable treatment methods that are effective there. Moreover, conventional treatment options are usually costly and impractical to conduct for small rural communities. The alga-based bioremediation process, or algoremediation, has been promising elsewhere but has not yet been widely researched. in this particular context. Chlorella vulgaris is the chosen algae species used in this method. This study is therefore required to assess the current condition of Pulau Seri Tanjung's water quality and look into the possibility of algoremediation as an environmentally friendly way to enhance the region's water resources. The objectives of this study are: first, to assess the current water quality status in Pulau Seri Tanjung, Tumpat, Kelantan. Second, to isolate and identify suitable local algal species for use in algoremediation. Third, to evaluate the effectiveness of algoremediation in improving water quality parameters such as turbidity, pH, dissolved oxygen, nitrate, temperature, and phosphate. The last one, to assess the water quality status by using Water Quality Index WQI and National Water Quality Standards NWQS as indicators. We move to the scope of study. This study will be conducted in Pulau Seri Tanjung, Tumpat, Kelantan. Water samples will be collected from 5 selected locations within the area and analyzed for biological parameters. Local algae species will be isolated and tested for their ability to remediate water quality in controlled environments. The study will focus on parameters such as pH, turbidity, dissolved oxygen, nitrate, phosphate, and temperature, with laboratory scale algoremediation trials conducted. Then, the significance of this study are expected to provide valuable baseline data on the water quality current status in Pulau Seri Tanjung, Tumpat, Kelantan, demonstrate the potential of algoremediation as an effective, low-cost, and eco-friendly method for water treatment in Pulau Seri Tanjung, Tumpat, Kelantan, then support local authorities, environmental agencies, and community members in implementing sustainable water management strategy. The last one, contribute to environmental conservation efforts by promoting nature-based solutions to address water pollution. For the literature review of this study, water sampling is a critical component in water quality assessment, serving as the foundation for evaluating the physical, chemical, and biological characteristics of aquatic systems. There are several approaches to water sampling, each designed to meet specific monitoring objectives. Water sampling is typically conducted to analyze a wide range of parameters. Physical parameters such as temperature and turbidity provide insights into the clarity and thermal characteristic of the water. Chemical analyses often include pH, dissolved oxygen, and nutrient concentrations. The health of local communities and biological system is influenced by the quality of the water, particularly in rural areas like Pulau Seri Tanjung, Tumpat, Kelantan. Pulau Seri Tanjung is a tourist attraction because there is a market that sells various types of goods. With the large number of people visiting the place, water pollution will occur due to their irresponsible attitude towards the environment. Therefore, regular monitoring practices and effective management development are required in this area to solve several issues such as anthropogenic activities, wastewater emissions from nearby towns, agricultural runoff from nearby fields, and possible coastal contamination. As Pulau Seri Tanjung is in the early stages of development, there is a limited amount of research available regarding the area. This study aimed to assess the water quality of the island and enhance it through the algoremediation process, which is claimed to be more cost-effective and energy efficient than conventional wastewater treatment methods. Algoremediation is the process of using algae to treat and improve the quality of polluted water by absorbing or converting contaminants into less harmful forms. In contrast, biological approaches have emerged as more environmentally friendly alternatives Among these, algoremediation, which involves the use of algae to remove contaminants from wastewater, has gained substantial attention. In this research, Chlorella vulgaris is used in the algoremediation process to treat water quality. Furthermore, in this research, the Water Quality Index (WQI) and National Water Quality Standard (NWQS) are used as indicators to determine the quality of water in Pulau Tanjung. WQI is one such method for evaluating the water quality. For the methodology, first, water sampling. Water samples will be collected from 5 locations around Pulau Tanjung using sterilized sampling bottles at a depth of 10 to 220 cm. Additional samples for algae isolation will be collected using an algae net. Then, samples will be stored in a cool box and will be transported to the lab within 24 hours for further analysis. Then, we will do initial water quality analysis. Samples will be poured into beakers and measured with cylinders. For the physical parameters such as temperature, pH, and turbidity were measured using a multi-parameter water quality meter. For the chemical parameters, nitrate and phosphate concentration will be analyzed using test kit. For the dissolved oxygen DO, will be measured using the dissolved oxygen or DO probe. Next is isolation and identification of algae. Algae samples will be collected from the water using an algae net and will be transferred to sterile containers. Samples will be serially diluted using sterile pipettes to reduce cell concentration and aid in colony isolation. Then, diluted samples will be stripped onto Petri dishes containing Bowls Basal Medium (BBM) under aseptic condition. Then, the plates will be incubated at 25 to 28 degrees Celsius with a 12-hour light or 12-hour dark cycle in an incubator for 7 to 10 days to allow for algal growth. Chlorella vulgaris will be identified as one of the dominant algal species, characterized by its spherical shape, non-motility, single cup shape, chloroplast, and solitary growth pattern. For algae cultivation, pure colonies of Chlorella vulgaris identified during the isolation process will be selected for cultivation. Selected colonies will be transferred into culture flasks containing sterile Bowls Basal Medium (BBM) using sterile pipettes. After that, flasks will be placed in an incubator with appropriate lighting to simulate optimal photosynthetic conditions. Cultures will be monitored daily for visible growth. Chlorella vulgaris cultures will be allowed to grow until they reach the exponential growth phase, typically within 7 to 14 days. For algoremediation treatment, actively growing cultures of Chlorella vulgaris in the exponential phase will be harvested for treatment use. Aliquots of the alga culture will be added to separate containers containing pre-collected polluted water samples. Each container will receive a consistent volume of algal culture, which is 10 ml. At the end of the treatment period, water samples will be collected from each container and analyzed to determine the efficiency of Chlorella vulgaris in removing nutrient and improving water quality. After the completion of the 14 days algoremediation period, treated water samples will be collected from each container. The same parameters that were measured during the initial analysis were re-evaluated. The last one, for data analysis, the result from day 0, day 7, and day 14 will be compiled and tabulated for each water quality parameter. Changes in water quality before and after treatment will be evaluated to determine the efficiency of Chlorella vulgaris in nutrient removal. Next, for the expected results, this table presents the expected changes in water quality parameters over a 14-day algoremediation process using Chlorella vulgaris. It shows that nitrate and phosphate levels significantly decreased by 69.6% and 73.2%, respectively, indicating effective nutrient removal. Dissolved oxygen increased by 41.7%, suggesting improved water quality. Turbidity decreased by 49.7%, reflecting clearer water. pH showed a slight neutral increase and temperature rose slightly by 4.3%, which is a normal variation during alga activity. Overall budget for this research is 960 ringgits, including apparatus, chemicals, and other expenses such as transport and field expenses. There are various sources of references that were needed as additional information to conduct this mini project. For example, additional information that was received from Google Scholar, Science Direct, Scopus, and more. That is all from me. Thank you.